let's let's continue our topic. We have discussed about the advantages and disadvantages of molten work. Now we will discuss about the discovery of peptide bond. In the peptide bond, the discovery take place, the natural product, the hormone in the human body. The history of the peptide bond discovery start the explosions, yeah, exploited natural product, hormones, peptide, well studied. So well studied physiological function of treating disease caused by the hormone deficiencies such as the lack of insulin required to regulate the blood glucose level in the patient with T1DM and T2DM. Diabetes is treated either by the insulin injection or by stimulating the insulin secretions related target such as GLP-1 and receptor to produce the insulin 48. Searching for natural peptides and the hormones replace them by animals homologies such as insulin, GLP-1, and somatostatin, GNR, and advosoprins and oxytocin were initial strategies used for the peptide drug discovery and the development. However, the drawback associated with the natural peptide interested optimize their natural sequence leading to series of natural hormone minima. So in this figure show that the structure of the human insulin and insulin drive drugs. Structure of human insulin is as the A chain A assigns the modification its residue and the resultant in the several short and long acting insulin drugs. Next, the topic is peptide mimicking hormone. GLP-1 drive peptide drugs, amino acid peptide regulate the insulin production and secretion. So in this figure, you can show that the insulin secretions, the chemistry structure, GNRF sequence of the structured natural hormones and GNRN and their the peptidomimetic drugs. GLP-1 drug, peptide drugs, is a 26th residue of natural sequence. Chemical synthesis of the peptide bond is the take place. The chemical synthesis of the peptide bond is well developed, particularly solid phase peptide synthesis technology developed by the Marified. The 1963 SPPS technologies has since been remarkably peptide production. It facilitates the peptide synthesis by combining amino acids and coupling the protections in one simple reactors. So as further led to be the invention of the automatic peptide synthesis compared with the combinant technologies. The crude peptide obtained by SPPS are more monotonous without other biological compounds such as enzymes, DNA, RNA fragment, non-related proteins, peptides. Moreover, the impurities in the final SPPS products are easily identified. They are mainly derived from the incomplete and side reactions during the synthesis of procedure 115, making sequence purifications uncomplicated. Then in this process, peptide, the general process of solid phase peptide synthesis will FMOC protect famous protected amino acids, famous double A OH, famous SPPS consists of cycle of coupling FEMO and the solid polymeric design protected of FEMO to liberate amino acids. So the development of chemical peptide synthesis, especially by SPPS, has significantly accelerated the development of therapeutic peptides. Some recombinant peptides, drugs such as cytosines, teriparatides, use chemical synthesis to produce the active pharmaceutical ingredient. So, chemical modification of the peptide and peptidomimetics, the particular class of the therapeutic agent biological activity peptide, is the intimately related to their chemical structures. So the backbone of the peptide bond in this figure, you can say that one of the main reason for the backbone modification is to improve the proteolytic stability of peptides. Proteolytic site in the peptide can be identified by the stability studies and metabolites determinations. Backbone modification include the substitutions of amino acids by D amino acids, insertion of the methyl amino acids, and incorporation of the beta amino acids peptides. Introducing these known amino acids into the peptide sequence, particularly at the proteolysis sites. So, conventional P glylations often occur lys and cis residue. So in this figure, you can say that I have explained about the genetic code expression. Expression provided the valuable tool for protein P gylations. One approach involves genetically coding C double A. So in the figure 10, we will see about the containing the bioorthogonal chemical handles at desired location into the target protein, followed by the conjugation PAC via the orthogonal reaction. In 2004, this will see that the diagrams I have explained that in 2004, dieters reported the first NCAC mediated mono P gylations method based on the genetics incorporation of P azidophenylaniline in the yeast. An alkyne derivatized PG chain was site specially attached to the superoxide dismutase, and the abbreviation of the dismutase is SOD via copper catalyzed alkyl azides cycloadditions click reactions with the PAZ, and the resultant SOD showed similar in the activity to Y type protein. ZHANG showed the mono-PC glycation. The example demonstrated by the mono-PC gylations can generally applicable to various proteins. The diagram show that these have the proteins like PAZ depth structures, neak structure, PACF, HIPPOC, P and para, like para, ortho, and a position described had para NO2F, and then 3 NO2 tire, SO3 tires. These are the further the example of the protein. Next, the scientist who got the Nobel Prize was K. Barry Sharpless. He was born on 28 April 1941, Palestine. Nobel Prize 2022, born 28 April 1940 in Palestine, USA, America. Scientists who are the co-winner of Nobel Prize for the chemistry in 2021 and 2022. He shared the 2021 prize with the William S. Knowles and Noyori Ryoji for developing the first chiral catalyst. With the Morten and Carolyn B. Bertozzi, he was awarded the 2022 prize for the contribution to click chemistry, bioorthogonal chemistry. Sharpless was just the fifth person to receive the two Nobel Prize. The work of Sharpless was that chemists tried to build increasing complicated molecules for a long time. He has been time consuming and expensive. Sharpless coined the concept of click chemistry, where the molecular building blocks snap together quickly, efficiently. 2002, Sharpless and the Morten Meldal independently of each other developed the elegant and efficient chemical reaction. The copper catalyzed azide alkyl cycloadditions. This is known in the widespread used in the utilization development of pharmaceutical for mapping the DNA creating new material. Research focus. Sharpless research focus on the chiral catalyst of the oxidation. Broad families of chemicals, atoms, or molecules undergo the oxidation and reaction lose electrons and certain their the functionality or capacity to form the chemical bond. In 1980, working at the MIT, Sharpless carried out the key experiment that led to the particular method based on the catalytic asymmetrical oxidation producing the epoxide compound used in the synthesis of heart medicine such as beta blocker and other products. The next scientist was the lady who was winner of Nobel Prize 2022 was the Carolyn R. Bertozzi. He was born on 16 January 1954. She was the Nobel Prize winner in 2022. She was the American chemist applicable for American chemist to study of biological systems. She coined the term bio orthogonal chemistry to describe the use of quick reaction, a simple chemical reaction to study the living cell. The work focused tried to build increasing complicated molecule for long. This has been very time consuming and expensive. Quick chemistry mean that the molecular building blocks snap together quickly and efficiently. Around 2000, Caroline Bertozzi started to utilize the click chemistry and living organisms. She developed the bioorthogonal reactions take place inside the human body. She has the graduated on the focus carbohydrates analog synthesis, the member of the Royal Society and academics of the science in the journal. The purpose of Bertozzi was organized for founding the field of orthogenical chemistry, the set of the chemical reaction that allow the research to study molecules, their interaction in the living things without the interacting with the natural biological process. The first development of the Bertozzi was the first lab experiment in the 1919 and early 2000s. Then had the lab and era have used to answer the fundamental question about the sugar in the molecule. Sugar high read more reflection. Bertozzi was awakened by the phone call from the Nobel Committee member who is in addition to breaking the mountainous news told her you have 50 minutes to collect yourself and wait until your life change. I could not be more delighted to Corais Bertozzi has won the Nobel Prize Chemistry due to the sugar high and read the more reflection from the colleagues' contribution to the chemistry. The next step of translating discovering in the recent years, Bertozzi said that she has grown increasing interest in applying the kind of experience chemist to medicine. In 2015, Bertozzi moved to the Stanford to help the Sarah Fan Camp, an institute devoted bridging chemistry. In 2020, he was named the Baker Family Director of Sarafan CAMH, and he was working to build strong connection between the foundation, chemistry and biological research, clinical rewards, research awards, and Stefan medicine. In her research, she has helped develop new lower the cost test for the tuberculosis, more efficient test for the HIV. Um, that's all from my side. That the he awarded many times Nobel prizes like protocolial training programs and the lecture Nobel edition, then and many other societies he gone to about the win the Nobel Prize. In this lecture, we have studied about the Nobel Prize who got the scientists that have worked in the chemistry field. So that's it from my side. Thank you.